everybody, and welcome back to the second episode of Bugs, Drugs, and Genomes. This is Ella,
Zephy,
and we're back for another episode from the Research Group for Global Capacity Building. Today, we're chatting with Natasia, a PhD student with the GloCab group, and she's going to help us break down the concepts of AMR, WGS, and share a bit about her work in the group.
Hi, Natasha. Welcome to the podcast.
Thank you. I'm so excited to be here today.
We're happy to have you here. Awesome. So, like Ella mentioned, you're a PhD student in the GloCab group. So, what does a PhD student exactly do?
So a PhD student is kind of a funny mix between being an actual student. So you take courses, you have deliverables, things like that. And then also working almost as a normal employee.
All right. Interesting.
And so what do you work on right now with your PhD?
So my PhD is about surveillance of antimicrobial resistance in low and middle income countries, mainly Africa, using a technology called whole genome sequencing.
So what is it exactly? What is AMR exactly for someone who has no background on AMR surveillance?
So I think the best way to think about AMR, which is an acronym for antimicrobial resistance, is that it's the way that bacteria or other pathogens can defend itself against antimicrobial agents. So kind of think of it as the sword and armor against antimicrobials. And it's becoming a big issue because, as most of people must have heard, you have an increasing amount of antimicrobial resistance against the antibiotics that people usually treat with in the clinical sector.
All right. It's becoming harder and harder to fight these bacteria or microbes.
And most people have been on some kind of antibiotic treatment and know there are several different kinds you give depending on which disease you have, but also sometimes it actually due to the one you took 5 years ago now has prevalent resistance. So if you treat with that antibiotic, you will have very little or no effect anymore.
And you mentioned that you work on Seek Africa. So does that mean you work with African countries on AMR surveillance?
Yes. Bacteria mutate at a much faster pace than us. So think of kind of an accelerated evolution against the natural environment. So obviously in places where you use antibiotic a lot more, or you don't necessarily use it at targeted treatment, you will find a much higher proportion of antimicrobial resistance. So that's one factor you have. That in countries where the population don't have a lot of money, if you go to the doctor and you have $5, you can either pay for the test or the antibiotics. You're going to give the antibiotic and not the test. While high-income countries, you usually see that you get some kind of diagnostic test and then the treatment. Another big factor that makes it antibiotic resistance a much bigger issue in LMICs is that you also have a lot more of what is called communicable diseases. So that's kind of like things like diarrhea and other diseases where people infect other people. So you see a large proportion of that in low and middle income countries. So that's also why it's so important for these interventions and surveillance to be also focused in low and middle income countries.
This sounds very, very interesting. It's so cool. And as a PhD student, what kind of data do you work on from these countries?
So my area of research is in something called whole genome sequencing.
All right. So for someone who doesn't know what whole genome sequencing is, what is it exactly?
So whole genome sequencing is when you take the DNA and then you kind of decode it. You know what exact, so DNA is of 4 different bases, and you kind of get the sequence. You get whatever the basis of DNA is. Whole genome sequencing is then when you sequence the entire genome. Other kind of sequencing, you can target specific regions if you only want to identify which pathogen or other type of thing. But whole genome sequencing, you can get kind of like all that would make you Zephy or you Ella. And I would be able to distinguish between who's who other kind of technology, I might be able to say, oh, Sefi is a human. But I wouldn't be able to say you're a female. I wouldn't be able to say you have black hair and other things that makes you unique.
Okay, that sounds so interesting. So from what I understand, you work on AMR and you also work on whole genome sequencing. So how does that go sort of hand in hand?
So I think the easiest way to understand how you can use whole genome sequencing in infectious diseases is to think about COVID, who we unfortunately
COVID, back to 2020. Yay!
Yeah. So we all have trauma for that time.
Absolutely.
But most of you might remember from the news that you heard a lot about some strain called Omicron.
Yes.
That was a specific kind of COVID that they could tell. And they also showed these maps where you saw how the Omicron version moved from Africa more north and how it spread. So that is actually what you can also do with antibiotic resistance. So you can distinguish in that way, but I can also track it because antibiotic resistance doesn't just move within the same pathogen. So we can have a disease outbreak with salmonella in food, right? Then you might track that specific salmonella and some of its characteristics. But bacteria has this amazing or terrible, depending on how you view it.
Depends on how it goes.
Ability to exchange something called mobile element and plasmid between each other. And you don't even have to be the same type of bacteria to exchange this information. So that means that when you do whole genome sequencing, you can actually sometimes track that exchange. And you don't have to have the same prerequisite to know what you're looking for.
Wow. That is so cool. Wait, so out of curiosity, so if I have E. coli, which is a type of bacteria, and then I have Pseudomonas. So from what I understand, E. coli and Pseudomonas can sort of exchange information and mutate either positively or negatively, and that depends on this exchange material, right?
Yes, exactly.
Wow. So whole genome sequencing helps to track this.
Yes.
Wow. Okay, that's very cool.
A family history.
And a
bacteria
map.
And a map.
Yeah.
Just out of curiosity, so if a DTU student decides to pursue a career in this field, what sort of background should he or she be from? And what do they need to have? What is the sort of skill set you need to have in order to pursue a career in AMR and whole genome sequencing?
So I think it depends a lot. My background is actually, I'm a civil engineer from DTU in biotechnology. So that is a bit of a different field, right? That's more towards using bacteria in food production. But you can also have other backgrounds. I was very interested in bioinformatics, which is kind of like the computer science behind all the sequencing data. And then it was kind of random that I ended up in the global capacity building group when I wrote my master thesis there. There's not any education that has AMR as like, this is the direction you can take your master's in, or like a subtopic. But yeah, so I think it's a lot about random opportunities.
And thank you so much for that wonderful overview. All right, everyone, thank you so much for tuning into another episode. But before you go, Zefi has a quick quiz question for us.
All right, let's do it. All right, so from what Natasha has explained so far, and so incredibly well, what do you think whole genome sequencing aims to determine? Your options are option A, the complete DNA sequence of an organism's genome, option B, measuring blood glucose levels, and option C, visualizing protein structures. And the answer is option A, the complete DNA sequence of an organism's genome.
Perfect. All right. Thank you so much, and we'll catch you for another episode soon of
Bugs, Drugs, and Genomes. All right. See you.
Bye.